Here are the details of some of the current clinical trials investigating gene therapy approaches for treating mesothelioma. Mesothelioma is a type of cancer that develops from the mesothelial cells that line the pleura, peritoneum, pericardium or testes. It is mainly caused by exposure to asbestos, and currently available treatments offer limited survival benefits. Gene therapy is a promising approach that aims to use therapeutic genes to directly treat mesothelioma or enhance the immune system's ability to fight the cancer. There are several ongoing clinical trials evaluating different gene therapy strategies for mesothelioma. One approach involves using an oncolytic virus known as Pelareorep, which is a modified reovirus. Reoviruses normally do not cause disease in humans. Pelareorep has been genetically modified to selectively replicate in and lyse cancer cells but not normal cells. It works by infecting and killing mesothelioma tumor cells, which then rupture and release new virus particles to infect neighboring tumor cells in a self-amplifying manner. This destroys the tumor from within. Pelareorep is also thought to stimulate an antitumor immune response. A phase 2 clinical trial investigated Pelareorep combined with chemotherapy in malignant pleural mesothelioma patients. Results showed the combination was generally well tolerated and appeared to improve progression-free and overall survival compared to historical controls treated with chemotherapy alone. Based on these promising results, a phase 3 randomized controlled trial, PROPEL trial, is currently underway to further evaluate Pelareorep combined with chemotherapy versus chemotherapy alone in previously untreated mesothelioma patients. The primary endpoint is overall survival. Another active gene therapy clinical trial involves an approach known as telomerase-specific replication-competent oncolytic adenovirus or TLRA. Telomerase is an enzyme that is normally active in stem cells and cancer cells but not in most normal cells, allowing them to proliferate indefinitely. TLRAs have been engineered to specifically replicate in and lyse telomerase-positive cancer cells. A phase I study assessed intratumoral injections of TLRAs for malignant solid tumors including mesothelioma. It showed antitumor activity with no dose-limiting toxicities. Based on these results, a phase IB/2 trial is ongoing to evaluate TLRAs alone or in combination with pembrolizumab, an immune checkpoint inhibitor, in mesothelioma and other solid tumors. Immune cell therapies that aim to enhance the body's natural anti-tumor immune response are also being explored. One such approach involves genetically modifying T cells collected from the patient to express a mesothelioma-targeting chimeric antigen receptor, CAR T-cell therapy. CAR T cells are a type of T cell that has been engineered in the laboratory to recognize and attack cancer cells. A phase I study conducted at the National Cancer Institute is assessing the safety and efficacy of administering autologous T cells genetically modified to target the mesothelin antigen on mesothelioma tumor cells. Mesothelin is highly expressed in many mesothelioma tumors but not most normal tissues, making it a promising target. Preliminary results indicate the therapy was well tolerated with some signs of anti-tumor activity. Based on these findings, further studies are likely to optimize this approach. Another immune cell therapy clinical trial involves using natural killer (NK) cells derived from umbilical cord blood, expanded ex vivo and genetically modified to express high levels of IL-15. Natural killer cells are a type of white blood cell that can kill abnormal cells like tumors. However, cancer evades NK cell-mediated killing through various mechanisms. By being augmented with IL-15, the NK cells are hoped to overcome these immune escape mechanisms and mount a powerful anti-tumor response. A phase I pilot study administered autologous NK cells to mesothelioma patients and found it to be safe and clinically feasible. Based on these results, further studies may investigate the most effective dosing and schedule as well as combining this therapy with checkpoint inhibitors. Gene therapy represents a promising strategy for mesothelioma, which currently lacks effective long-term treatment options. Several approaches including oncolytic viruses, tumor-targeted immune cell therapies, and gene-modified cell therapies are currently being evaluated in clinical trials. While still in early research stages, some have shown safety and preliminary signs of anti-tumor activity. With further optimization, these gene therapies may potentially improve survival outcomes for mesothelioma patients by directly targeting the tumor or enhancing the innate immune defenses against cancer. Continued clinical research is important and likely to yield more efficacy data in the coming years.